me next Tuesday along with a new problem set. And so one other note, I've gotten a couple of questions about the fact that the problem set has a number of problems on DNA topology, and that isn't because that's going to be sort of overrepresented on the exam or anything. It's just it seems to be a topic particularly confusing, so I wanted to make sure that people had a number of questions they could practice on and discuss. Yeah. So yeah, the question is whether the answer key could be posted earlier, and actually because some people will be having their quiz this week already, in the interest of fairness, we're actually not going to post the key at all until everyone has taken this round of quizzes.
Did you have toes this early? Did you click them off? Wednesday? That's the one I'm trying for.
So
it's at night, so
today I'd like to start discussing DNA replication. So this is one of the sort of the three basic steps of the central dogma to tell us that genetic information is propagated by DNA replication, and then that information in the DNA, of course, is transcribed and translated to make protein. And so replicating the DNA faithfully is extremely important for the inheritance of the genetic information. And so we're going to start by talking about the fundamentals of DNA polymerase. We talked a little bit about some of the reaction developments before. And that will give us a picture of sort of locally how you can copy a sequence of DNA. When you think about copying not just a short piece of DNA, but in fact copying an entire genome, there are a few other problems that come up, and we'll discuss how the cell establishes what are called replication forks, in which both of the strands of DNA are copied simultaneously, and how the cell achieves the sort of processivity, that is how it actually successfully runs through an entire, in the case of E. coli, an entire over 2 million bases of DNA copying. And finally, we'll talk a little bit about the processes of initiation and termination, mostly about initiation and sort of, again, just managing some of the problems, including some problems of DNA topology that come up in copying an entire, either an entire circular chromosome or an entire linear chromosome in eukaryotes. The basic reaction of DNA polymerase is the same trans-esterification reaction we've seen before. So in DNA polymerase, there's a template strand, it's a pre-existing strand that's being copied, and a growing primer strand. So DNA polymerase can only extend a pre-existing nucleic acid strand, and it extends it always at the 3' end with a DNTP that's complementary to the base on the template strand. And the mechanism here is very similar to the mechanism that we always see in the nucleic acid backbone chemistry. In this case, there are two magnesium ions that play an important role, and what they allow is a nucleophilic attack by the 3' OH of the growing strand, the primer strand, onto the alpha phosphate of a DNTP. And that ends up displacing the inorganic pyrophosphate, and that provides the energy to drive this reaction forward. And then, so what this means is that it's always the growing strand, the primer strand, grows from its 3' end by adding another nucleotide. It incorporates the alpha phosphate from a DNTP into the backbone and gets rid of the beta and the gamma phosphate. And so the DNA polymerase always is extending the 3' end, and after it does this insertion, it can translocate, that is, the nucleotide that it just added, that new nucleotide also has a 3' OH, and so it's a new substrate for DNA polymerase. And so the polymerase moves one nucleotide down and then repeats the same reaction. And processivity is the term for the number of times the polymerase typically repeats that cycle of adding a nucleotide and then moving one step down before it falls off the DNA. Of course, there are two competing processes. One is that it could translocate and then add a new nucleotide. The other is that it could simply dissociate. And of course, if you're copying a large genome, it's important to have high processivity so that you can actually make it all the way through that genome. was the other important point, and this is particularly important for DNA polymerase, is that it has to have high fidelity. That is, it has to only incorporate AT and GC base pairs. It has to be very specific for those base pairs, or it's going to introduce mutations into the DNA. And so the polymerase uses steric constraints to ensure that only the correct base pairs can be incorporated. So of course the AT and the GC base pairs are the energetically most favorable base pairings that can happen in DNA, but there are other pairings that can occur. For instance, G and T can mispair in a way that's not as energetically favorable as G and C, but is actually still pretty reasonable. And a number of the bases can undergo tautomerization, in which there is a sort of a low frequency alternate configuration of the base that can mispair. And so DNA polymerase has extra mechanisms to reject any of these mispairings resulting from this GC wobble pairing or these tautomerizations, and instead accept only AT and GC base pairs by essentially having the active site of the enzyme is spatially constrained so that none of these alternate pairings can form. And as a result of this, the error rate in simply the addition of nucleotides to the growing chain is somewhere between 10 to the minus 4 to 10 to the minus 5. So 1 out of every 10,000 to 100,000 nucleotides that gets added is actually a mispairing. So this is quite good fidelity, but it's not actually good enough to replicate the entire, even E. coli genome without introducing quite a lot of errors. So there are over 4 million bases in the E. coli genome. So there would be dozens or even hundreds of mutations introduced every time the DNA was copied if it relied simply on the fidelity in the incorporation of the DNTP. Yeah. Yeah, so the question, which is an excellent question, is that we talked before about how you might have chemical changes in the template strand, and that when the DNA polymerase encounters one of these chemically modified bases, that chemically modified base can mispair. And the answer is that yes, in some cases, that chemically modified base will cause a mispairing and actually cause a permanent change in the DNA sequence. And in other cases, these steric constraints mean that even though the base could pair in sort of normal DNA, in the context of the polymerase active site, no nucleotide will pair after the polymerase will just stop. So both of those things can happen, mutations or the polymerase actually stopping. And in fact, in the next class, we're going to talk about the fact that the cell has many different mechanisms to repair DNA, and the importance of those is exactly that if you have that damaged base, it is eventually converted into a normal base pair that's mutant in sort of in the long run. Yeah. Yeah. I mean, the overall, fidelity is just, it's a measurement of how often the polymerase incorporates the wrong base. And so the fidelity of the polymerase active site itself that adds the NTPs is this number. It's about 1 in 10,000 to 1 in 100,000 errors. But actually, all DNA polymerases in the cell have a proofreading mechanism. All the replicative polymerases in the cell have a proofreading mechanism that can catch many of these errors after the nucleotide is incorporated. So it's after the phosphodiester bond has been formed, but before it moves to the next step. Because the phosphodiester bond has actually been formed, this means that the mispaired base actually has to be cut off, and that the polymerase essentially has to try again. And this proofreading mechanism catches over 99% of misincorporation errors. So essentially, if you're going to add a nucleotide, about 99.99 or more % of the time, you simply add the correct nucleotide. Occasionally you have the wrong nucleotide. This happens less than 0.01% of the time. When that does happen, you have at least a 99% chance of cleaving that off and going back to try again, and less than a 1% chance that you'll leave that error in and then add the next nucleotide. And so the combination of this improved fidelity from proofreading and the intrinsic fidelity of adding DNTPs gives you a final error rate that's good enough to replicate the E. coli genome with essentially no mutations. And so the ability of the polymerase to cleave a phosphodiester bond as well as to form it means it has two separate enzymatic activities on two separate sites. And the process of proofreading is shown here, that when there is a misincorporation, here they're showing a CT misincorporation, the translocation is slowed. The polymerase sits on top of that misincorporated base for longer. And there's a sort of an alternate reaction path where the polymerase can reposition that mispaired base into a separate exonuclease site. So this is a 3' to 5' exonuclease, meaning it's going to cleave off the nucleotide at the very 3' end of this bearing strand, which is exactly the one that just got misincorporated. And so this separate nuclease site uses, again, this nucleophilic attack on the phosphodiester backbone to remove the bond that the polymerase just made. And it goes back and tries again to incorporate the correct base. DNA polymerase itself, there is in E. coli an enzyme, DNA polymerase 1, that has both this DNTP addition reaction and this exonuclease proofreading reaction. And so this was the first this DNA polymerase 1 was the first polymerase isolated from E. coli. It was isolated by Arthur Kornberg in a pretty classic sort of biochemical approach where he found that there was an activity in E. coli that could incorporate radioactive DNTPs into DNA. And then he would make E. coli extracts and fractionate those until he found that there was actually a single polypeptide, its structure shown here wrapped around DNA, that was capable of doing this DNA polymerization reaction. So it's an abundant single subunit protein in E. coli. And the real structure is shown here. The schematic structure that we use a lot is shown here. It sort of curves around the DNA, and sometimes it will make an analogy to the shape of a hand wrapping around the DNA. But it turns out that there are some quantitative discrepancies between how DNA polymerase 1 synthesizes DNA and the actual replication of the whole E. coli genome. Essentially, it's too slow by over an order of magnitude to actually copy the E. coli genome as quickly as we see that done, and it's not processive enough, and then it falls off often. And maybe the most serious concern is that mutants in E. coli were isolated that no longer made this DNA polymerase 1, and they were capable of replicating their genome and dividing just fine. They had some problems. They were a little bit sick, but actually not that sick. And so it turns out that DNA polymerase 1 plays an important role in replication. It's not the major replicative polymerase in E. coli. I guess now I'd like to turn to some of the broader questions about how you replicate a whole genome as opposed to how you simply sort of synthesize small pieces of DNA. And one very early and important observation about this was made in this Michelson-Stahl experiment, in which it was shown that the process of DNA replication overall was semi-conservative. And so what that means is that if you have the pre-existing template strand, and then you look at the two daughter double helices, what you'll find is that each daughter double helix contains exactly one old and exactly one new strand. So it isn't the case that, for instance, you copy the double helix and you take both new strands and zip them together and keep one entirely old double helix around. And it isn't the case that it's large, sort of a mixture either, that each strand is sort of a mix of old and new sort of stuck together. You can follow these old and new strands, and in this experiment, they used labeling with a stable isotope of nitrogen that's heavier, and so DNA containing this heavy nitrogen is slightly denser, and you can separated out. So what you find is that the original parent molecule contains entirely heavy DNA, and so it's very dense. But after the first generation, all of the daughter molecules contain one old and one new strand, and so they migrate at this intermediate point. And then after the second generation, some of the molecules still have one entire old and one entire new strand. These are remain in this hybrid fraction, and then the others result from a first generation new strand being copied into a second generation new strand, and so they're 2 generations entirely alike. And so this is sort of the phenomenon that you can see molecularly. You can also actually watch some of this process by electron microscopy, and here's a picture of replicating E. coli genome caught at different points during replication. And so because the E. coli chromosome is one large circle, and the replication is happening at these 2 what are called replication forks. And at each of those points, both strands of the genome are being copied. So this sort of spreads around until eventually it forms this structure that's sometimes called a theta structure, because it looks like the Greek letter theta, until eventually you have 2 almost entirely separate strands, and eventually you do some tricky stuff right at the end to separate these 2 circles. Elongation at these 2 replication forks starts and travels all the way around the circle, meaning each fork replicates over 2 million base pairs of DNA. Here's just sort of a note about this quantitative discrepancy about polymerase I being too slow and falling off too often in order to actually replicate the genome. And so the question is, how do you make a DNA polymerase that's more processive and that doesn't fall off? And the answer is essentially that you lock the DNA polymerase physically onto the template strand. So DNA polymerase III is the major replicative polymerase in E. coli. It's not a single protein, it's actually a pretty large complex. And one important feature of that complex is called the beta sliding clamp. So this is a homodimer of 2 proteins. One of them is shown in dark purple and one in light purple. And they physically wrap around the DNA and hold the polymerase on. So the reason it's so processive is that it's physically linked to the DNA and it can't fall off. So overall, the replicative polymerase has this beta sliding clamp that's attached to the polymerase III core. So this is really the active sites of these enzymes, the alpha subunit. So each of these Greek letters represents a distinct polypeptide chain. So all of these proteins assemble this large complex. The alpha chain contains the actual polymerase activity that does the transesterification of a DNTP onto the growing 3' end. The epsilon subunit contains a proofreading activity, so in this enzyme you can actually separate the addition of DNTPs from the removal of misincorporation. There's a beta sliding clamp, and then there's this structure here that holds 2 polymerases, 2 polymerase cores together, and also has the ability to load and unload this clamp. Of course, if you're going to lock the polymerase onto the DNA, at some point you need to open up that clamp, put the DNA through it, and close it back up. And so this clamp loader is responsible for that activity of opening the beta clamp. And so you can see that this whole Pol3 has actually 2 sort of active Pol3 cores, one to replicate each of the 2 strands of the DNA. And so both strands of the DNA are copied together at the replication fork, and this leads to a slight problem in that so the replication of DNA is always 5' to 3' on the strand that's growing. Of course, the double helix is anti-parallel. And so what that means is that there's one strand where the polymerase can just go 5' to 3' easily and continuously in the direction of replication as the fork grows. The other strand, however, is trickier because 5' to 3' would mean you'd want to synthesize DNA this way. That's the direction of synthesis, but the whole fork is moving this way. And so this problem of the simultaneous replication of anti-parallel strands. Which way does this DNA polymerase go? The problem, so this is called the leading strand, is the one that's synthesized quickly, and, easily, and continuously. And this lagging strand has this problem that the polymerase moves this way in each individual step, but its net movement is in this direction. And so the solution to lagging strand synthesis is sort of, I think, illustrated best by this video. It's sort of an artifact of the 1980s. But let's see if this actually works. The DNA polymerase is played here by Michael Jackson. And you can see that he takes individual steps that are 5' to 3' strands being synthesized, but the net movement of the polymerase is the opposite direction. This is exactly what DNA polymerase does. It takes a series of short 5' to 3' steps on the lagging strand, but the net movement of the polymerase is in this other direction on the replication fork. And so these short fragments are called Okazaki fragments, after the person who first identified that the lagging strand is actually synthesized in a sort of discontinuous way. And each of these Okazaki fragments, so in E. coli, these are 1 to 2 kb long, and eukaryotes are actually even shorter, that the polymerase is taking steps of just a few hundred base pairs each time, is synthesized away from the progressing replication fork, and the polymerase moves back and does another one, and so forth. And so one problem that is, a problem that does arise for the leading strand, that's synthesized continuously, but it's a big problem for the lagging strand, is that I told you that polymerase can't actually start a totally new DNA chain. Polymerase can only extend an existing chain, and especially for these Okazaki fragments, that means something needs to start the chain at the beginning of each of those Okazaki fragments. So the problem is that DNA polymerase can only extend. In fact, this is true of all DNA polymerases. And so there's an enzyme called primase that actually synthesizes an RNA primer that begins well, first of all, it begins the leading strand, but more importantly, it begins each of these Okazaki fragments. This primase enzyme will synthesize a short, I think about 20 base of a short RNA primer, and then the DNA polymerase can extend either an RNA or a DNA. It's perfectly possible as a molecule. It's a hybrid of RNA and DNA bases. The RNA is a perfectly good 3' OH that the DNA polymerase is happy to initiate on. And so this repriming by primase has to happen at the beginning of each Okazaki fragment. Of course, this introduces actually a new problem, which is that DNA is actually now full of short RNA primers. That not only is the lagging strand broken up into 1 kb chunks instead of being a continuous piece of DNA, that actually the beginning of each of those chunks is RNA, not DNA, and you definitely don't want to leave those RNAs in the genome. And so in a minute we'll talk about how the cell deals with that problem. I'll just sort of put together a picture of what's happening on the lagging strand. This illustration that, there's a helicase, a DNA helicase, that's unwinding the strands of the DNA at the beginning of the fork. And right next to it is this primase that's synthesizing an RNA primer. So on the top strand, the leading strand, the polymerase can synthesize all the way up to the fork itself. But of course, on the lagging strand, there's a region of unsynthesized single-stranded DNA, and the cell actually protects this unsynthesized single-stranded DNA with a single-strand binding protein that's coating the lagging strand until it's replicated. So the primase creates an RNA primer, and then at that point, the lagging strand, Pol3-4, can load on to that primer and start extending it, and it will displace the single-strand binding protein until eventually it runs into the RNA primer and stops. And so this is the synthesis here, and then here you're back essentially at the same place you were here. The leading strand is all the way up to the fork. The lagging strand has primase at the site of the fork synthesizing a new primer that eventually Pol3 will extend past those SSBs. Of course, another problem with lagging strand synthesis is, I told you that the polymerase has this clamp that gives it amazing processivity, where it can go hundreds of thousands of base pairs without falling off, but of course the lagging strand polymerase actually does need to fall off every time it runs to the end of one Okazaki fragment and needs to take one of these big steps backwards. And the answer is the baiting slided clamp is actually just left behind on that Okazaki fragment. The clamp loader will remove that clamp and then add a new clamp to Pol 3 at the beginning of the next Okazaki fragment at that primer. The leading strand baited clamp never needs to dissociate, and so it remains and tethers the entire Pol 3 complex to the replication fork and gives it sort of processivity it needs to follow the genome all the way through millions of bases in replication. And then this final problem that I mentioned is how to finish the lagging strand. So after the fork has passed, we're left with an RNA primer extended into Okazaki fragment, the other one behind it. So first of all, there's a break in the phosphodiester backbone. DNA polymerase doesn't link the strand it's making to the 5' end of that RNA primer. It can't make that kind of bond. Furthermore, there's a chunk of RNA in there that we need to get rid of. And so there are two enzymes that work together to do this. The first is DNA polymerase 1. So this is actually the function of DNA. One of the most important functions of DNA polymerase 1 in the cell is to clean up these RNA strands left in the genome. And it actually has the ability to bind to a nick site like this and chew up RNA or DNA in front of it while it synthesizes RNA or DNA behind it, which is a perfect activity, of course, to chew up this RNA and replace it with DNA. It still can't form this final nick sealing reaction, though, and so there's a separate enzyme called DNA ligase that uses energy to close this nick. Of course, for adding DNTPs to a growing chain, you have the energy of the DNTP itself to drive that forward. And energy isn't available to simply close a nick. You're not adding any new DNTP, and so DNA ligase needs to use energy to drive that reaction forward. So the first step of this, I said, is carried out by DNA polymerase 1. It has a unique 5' to 3' exonuclease activity. So the proofreading activity of DNA polymerase lets it back up, lets it go 3' to 5' by removing misincorporated nucleotide. And this is an exonuclease activity that chews up one DNA strand, but it moves in the opposite direction, and it's sort of out ahead of the active site of the polymerase, removing whatever is in front of it. So the 5' to 3' activity goes 5' to 3' on this Okazaki fragment, whereas the polymerase activity goes 5' to 3' synthesizing a new strand, and then eventually, so it extends this sort of later synthesized Okazaki fragment out through the RNA primer of the earlier Okazaki fragment. As I mentioned, leaving the single place where there is a break in the phosphodiester backbone, and that's what DNA ligase is responsible for handling. So DNA ligase performs actually 3 of these transesterification reactions. The first is that the enzyme itself has a nucleophilic nitrogen that attacks an alpha phosphate, either of AMP or of NAD, depending on the specific enzyme involved, but ends up with this situation where the enzyme itself is actually conjugated to the phosphate of this AMP. It's called adenylation. There actually are supposed to be 2 YLs there. And then the enzyme AMP conjugate actually adds to the 5' phosphate, left 5' monophosphate that's left behind at the site of the nick in the DNA. So basically one of these oxygens attacks the phosphate on the enzyme and then releases this enzyme nucleophile, and now you have a pyrophosphate here. And this is something that has sort of the energy that allows it to actually drive this ligation reaction forward. And the final step is that the 3' OH of the later Okazaki fragment attacks this phosphate, this leaves, and you've actually sealed the nick, and now you have a bond from the 3' OH, the later Okazaki fragment, back to the 5' phosphate on the earlier Okazaki fragment. So none of the phosphate groups from this AMP or this NAD actually end up in the DNA, but this enzyme does finish the DNA strand to ensure that you have a continuous DNA strand. So sort of all of these things together are a structure called the replosome, which is the DNA polymerase itself and all of these auxiliary factors that all travel along the replication fork. And so here, sort of a question to think about all of these different molecules involved. Give you about a minute and a half to talk about this. The question is, of all these things in the overall replosome, polymerase 3, polymerase 1, ligase, primates, and helicase, which of these does not form phosphodiester bonds in the course of synthesizing the new strand? Which does not?
C, D.
Sorry.
Okay, good. Wrap up in the next few seconds.
Today's not presentation.
Okay, and go ahead and stop now. So, DNA helicase is the one enzyme that does not form phosphodiester bonds. So the polymerases, certainly Pol1 and Pol3 are both forming some of these bonds that end up in the final DNA. Ligase does as well, to form that one last phosphodester bond that ends up in the DNA. Primates forms RNA-DNA bonds. None of those actually remain in the DNA backbone, but it is an RNA polymerase, and so it forms phosphodiester bonds in the RNA backbone. This DNA dehelicase does not form any of these bonds. Its role is actually to separate the two strands as the replosome moves forward. So, yeah. Yes, the question is, why does primates add RNA instead of DNA? I don't know of a really clear answer to that question. There may be some sort of, essentially, cellular defense mechanism that the cell never wants to start synthesizing DNA totally de novo as a way to protect it from foreign DNA. That's one possible explanation. I don't think there's a really clear explanation of that. So the process of replication initiation in bacteria actually begins with loading of this DNA dehelicase that's going to melt apart the fork. And so it really coordinates all of the events that are going to happen in the assembly of the replicative polymerase at the replication fork. And the process of replication initiation actually depends a little bit on some of the DNA topology that we were talking about. There's a single specific sequence in the E. coli genome where replication begins. This is called ORIC for origin of chromosomal replication. So origins of replication are the places where DNA replication begins, and it doesn't just sort of begin randomly. Really, in any organism, it doesn't begin totally randomly. And in bacteria, there's usually one specific site on the whole circular chromosome where it starts. And there is an ATPase called DNA-A that binds to the DNA at a number of these DNA-A binding sites at the origin of replication. And when it does that, it actually wraps the whole double helix around itself in a manner that's sort of analogous to the way that histones do this. So it's not a related protein to histones, but the manner of this wrapping is sort of similar. And so what that means is that you're introducing, in this case, the DNA-A is wrapping it in the other direction. And so it's actually introducing positive supercoiling in the DNA, positive solenoidal supercoiling, as the DNA wraps around it. And so that means that the rise of that DNA molecule is going to increase from this solenoidal wrapping. Now this whole thing is in a sort of a topologically constrained domain, so the linking number can't change. There are two other things that could happen. One is that you could get compensating negative plectonemic supercoiling, but the other that can happen at this specific region is that you actually get under-twisting of the DNA. So the linking number has to remain the same. As the rise goes up from this positive solenoidal supercoiling, the twist goes down, and this special sequence is actually relatively easy to untwist and melt apart the two strands over a region of almost 40 bases. These DUEs are a sequence that happens to melt easily. You can see they're very rich in A's and T's. And into this separated region, the replicative helicase loads on to the beginning of the replication fork. So the replicative helicase is also a ring that encloses one strand of the DNA. It's actually a hexamer of this protein DNA-B, and again, because it normally entirely encloses a strand of DNA, it has to be specifically loaded. And in this case, this DNA-C is a loading factor that uses ATP to open that up and then lock it back around the strand of DNA. And so once it's locked on, it'll establish a replication fork. It'll bring in primase. Primase will synthesize RNA primers, the leading strand. Polymerase will load on and will keep going all the way around the circle. The lagging strand polymerase will also be brought in and it will start synthesizing Okazaki fragments all the way around until these two divergent forks end up meeting at the end. So there's an origin here, and there are some terminator sequences here, but mostly what happens in bacteria is that the two forks moving in opposite directions simply run into each other. So you have a counterclockwise moving fork and a clockwise moving fork. And of course, as these two things are moving together, there's a lot of topological strain being built up here that has to be relieved by topoisomerases. And then actually, we don't totally understand what happens at the end when these two forks run into each other, and it's somehow resolved in a fairly clean way. But what is clear is that you end up with what are called catenated chromosomes, where one of these huge circular chromosomes is actually interlinked with the other. And this has to be resolved by a type 2 topoisomerase, or when the cell goes to segregate its chromosomes, there's physically one linked around the other and can't be pulled apart. So this role of the type 2 topoisomerase in concatenating both big E. coli genomes as well as plasmids is important to remember that the type 2 topoisomerase is the kind that can actually break both strands of a double helix and pass a separate double helix through it. So while a type 1 topoisomerase can relieve the strain that builds up as these forks move towards each other and sort of twist the DNA up, only the type 2 topoisomerase can actually separate the two different double-stranded molecules when they're done replicating. Yeah? So the question is whether type 2 topoisomerases require energy in their reaction. So type 2 topoisomerases do use ATP, but the net energy in the reaction is zero, because so it turns out that they use ATP probably to ensure that the reaction goes in one direction, but the sort of energy balance in the end is just that energy is sort of spent making sure the reaction goes in the right way, because you break the backbone and then you reform it. And so what actually happens is that there's a nucleophile in the enzyme. This is a tyrosine with an OH that attacks the phosphodiester backbone, holds on to it. It's very important, especially when you're breaking both strands. It's very important that you don't let go of that break, because otherwise I mean, the topoisomerase is the only thing holding that double-stranded break together in the cell. The second strand passes through, and the topoisomerase transfers the phosphodiester bond back off of the tyrosine onto the other side of the break. So replicating a eukaryotic chromosome is similar in many ways to replicating a bacterial chromosome, at least sort of locally. It's the process of copying both strands at the same time at a replication fork. Because, in part of the topology and in part because of the size of the eukaryotic chromosome, there are some differences. Just showing that even at the maximum speed of a bacterial Pol III replication fork, it would still take much too long to actually copy an entire human chromosome. If you had only one replication fork going, it would take something like 3 days. In fact, eukaryotic linear chromosomes are replicated from several places at the same time. They have many origins of replication, places where replication begins, and you get these replication bubbles with the fork at each end moving away from the origin of replication. These spread out eventually. These two replication forks run into each other, complete replication, and there's some ligation that happens to connect these newly growing strands, and eventually end up with two completely replicated daughter molecules. So I said that the eukaryotic replisome is actually quite similar to the prokaryotic one. I just put another picture of the prokaryotic one up here and labeled a few of the things with their eukaryotic names. The main replicative polymerases in eukaryotes are called DNA polymerase, delta, and epsilon. So it's slightly confusing because the individual subunits of the E. coli polymerase 3 are named with Greek letters, whereas different eukaryotic polymerases are named with Greek letters, for instance, alpha and so forth. And it turns out that delta and to some extent epsilon are entire polymerase complexes that are replicative in eukaryotes. The replicative helicase is called MCM 2 to 7. It's called 2 to 7 because, in contrast to the bacterial case where it's 6 identical subunits, in this case it's 6 different but related subunits. So we often see this that in bacteria there's a 6-member complex with 6 copies of the same thing. In eukaryotes, that gene has been duplicated 6 times. They've been slightly specialized, and so you assemble this ring with 2, 3, 4, 5, 6, and 7 subunits all around it. The processivity clamp is called PCNA, which stands for proliferating cell nuclear antigen. It turns out that this clamp is present only in cells when they're replicating their DNA, and that's where this name comes from. It turns out that PCNA is a trimer and not a dimer, but the basic idea that it's a clamp that physically encloses the DNA and keeps the polymerase on the DNA so it has high processivity is still the same. In eukaryotes, replication initiation is in some ways actually fairly similar. In other ways, it's regulated differently. So because eukaryotes have many origins of replication scattered all across their chromosomes, it's very important to ensure that each of those sequences, each of those origins, triggers replication exactly once. And so there's this process called licensing that happens before the in G1, so this is before the cells begin DNA replication. They assemble the origin recognition complexes onto origin DNA. And so they assemble in G1, and then on the entry to S phase, the cell changes so it's no longer possible to assemble a new origin, but the assembled fork complexes can can trigger a round of replication initiation. So they do this, in fact, by using ATP to unwind DNA at the origin site and to load on the replicative helicase. So like in bacteria, the first step in replication initiation is unwinding the DNA and loading on the replicative helicase is going to establish the whole replication fork. The process is somewhat different, though, in that you assemble this origin recognition complex that's going to load that helicase in G1. You trigger it to work exactly once at the onset of S phase, load on that complex, and then replication can proceed. So in yeast, there's a specific sequence motif that defines a yeast replication origin. It's called an autonomously replicating sequence, simply because if you put one of these sequences into a random piece of DNA and put it into a budding yeast, the budding yeast will copy that DNA as it enters S phase. So it allows the piece of DNA to be autonomously replicating in the cell. And there are about 400 of these ARS sequences across the 16 chromosomes, the 12 megabase genome of yeast. So obviously the number of ARSs per chromosome is much higher than in bacteria. So vertebrate origins, it's clear that there's a stereotyped place where replication tends to initiate. At this point, people have some pretty high-resolution maps of that process, but it doesn't seem to be defined by specific sequences. It's not entirely clear how it is defined in vertebrates. But we do know that between yeast and vertebrates, this process of assembling an origin recognition complex in G1 and having it fire to load MCM2 to 7, the replicate of helicase in S phase, is conserved. We just don't know why ORC assembles in certain places. So the other problem with replicating a linear chromosome, especially on the lagging strand, is what's called the end replication problem. So after you replicate a piece of DNA, on the lagging strands, the very ends are left as RNA primers and nothing else sort of further out on the sequence. And this is a problem because what that means is that when you get rid of those RNA primers, you actually have a gap, and you have no way, because DNA synthesis always has to go 5' to 3', you have no way to fill in that gap. And so that means is that you do this, you leave those gaps, and if you did another round of replication, you'd actually start to erode the chromosome further and further back in the end, and you start losing genetic information off of the ends of chromosomes. And this is a problem, and eukaryotes solve this problem by placing a special structure called a telomere on the end of these linear chromosomes. So telomeres are a repetitive sequence with no sort of important genetic information in them. They're synthesized actually from an RNA primer by this enzyme called telomerase. And they're just many, many repeats of this TTGGGG sequence, at least in humans. It's copied by, that's added by telomerase, and so you still have this gap left by primer removal when polymerase replicates to the telomeres, but it doesn't matter because there's no useful information here, and telomerase can go back and re-add a new sequence to the ends here. And so this is how eukaryotic cells solve the end replication problem. Another important point is that telomerase extends these ends, and actually in many cells in your body, telomerase is actually shut off. And so what that means is that as those cells go through many rounds of replication, their telomeres erode, and eventually their telomeres erode all the way, and that's the point where actually those cells stop dividing. And the reactivation of telomerase is one important point in sort of checkpoint in controlling the development of cancer. The cell needs to do something, either reactivate telomerase or find some other solution to these DNA ends in order to successfully replicate indefinitely. So this is sort of an interesting bit of molecular biology that's turned out to have a pretty profound impact on sort of cell biology and on preventing cancer in a multicellular organism. So that's what I have to say about DNA replication, and talk about repairing.